Describe the structure of HIV and laboratory diagnosis of HIV infection. Structure of HIV मा के हुन्छ भन्दा सबभन्दा भित्र स्ट्रक्चर अफ HIV मा सबभन्दा भित्र हामी हेर्न सक्छौं रिभर्स ट्रान्सक्रिप्टेज जुन चाहिँ कोइल जस्तो फोल्ड भएको छ अनि डल्लो यो ट्रान्समेम्ब्रेन ग्लाइकोप्रोटिन GP41 ट्रान्समेम्ब्रेन ग्लाइकोप्रोटिन GP41 डल्लोको बाहिरको भाग अनि त्यही फोइल भित्रै इन्टिग्रेज भनिदो रहेछ डल्लो भित्र फेरि सर्फेस ग्लाइकोप्रोटिन GP120 सर्फेस ग्लाइकोप्रोटिन GP120 अनि यो कोर सेल अल क्याप्सिड P24 कोर सेल अल क्याप्सिड P24 अनि फेरि आरएनए जिनोम एसेट्रा अब जान्छौं डेफिनेसन व्हाट इज HIV ह्युमन इम्युनोडेफिसिएन्सी भाइरस HIV is a virus which targets the human immune system of a person. With time, this HIV weakens and eventually damages the immune system of the individual. The body starts to lose the ability to defend itself against germs, bacteria and infections. Hence, it is difficult to fight this disease. Structure of HIV. Approximately, HIV has a diameter of 100 nanometer. It has an envelope of lipids which embeds the GP41 trimeric transmembrane glycoprotein. To this, the GP120 surface glycoprotein is attached. Both proteins are involved in attaching to the host cell. It is encoded by the ENV gene of the RNA genome of the viruses. Core proteins P6 and P24, P17 matrix protein and P7 nucleocapsid protein, all of which are encoded by the viral gag gene, are seen underneath the envelope. There are two copies of the 10 kilobase kb positive sense viral RNA genome, together with the integrase, protease and enzymes reverse transcriptase in the viral code. The viral pole gene encodes for all these enzymes. There are numerous other proteins that are coded by HIV1 and HIV2 with different immunomodulatory or regulatory infections such as VPR, viral protein R, VIF, viral infectivity protein, NEF, negative regulatory factor, REV, regulator of viral protein expression, and TAT, transactivator of transcription. There are numerous other proteins that are coded by HIV1 and HIV2 with different immunomodulatory or regulatory functions such as VPR, viral protein R, VIF, viral infectivity protein, NEF, negative regulatory factor, REF, regulator of viral protein expression, and TAT, transactivator or transcription. HIV1 comprises two copies of non-covalently associated positive sense single-stranded RNA encased by a capsid comprising P24 viral protein. Both copies of RNA strands significantly contribute to the HIV1 recombination occurring at the time of reverse transcription of the viral replication. Pseudodiploidy is when two copies of single-stranded RNA are contained inside a virion. However, only a single DNA provirus is produced. The long open reading frames ORFS encode the viral structural proteins, while the smaller ORFS encode the regulators of the viral life cycle. Microbiological laboratory diagnosis. OI diagnosis, PCR, RT-PCR. सुरुमा छ direct detection culture. The detection of HIV1 by mixed lymphocytes or co-culture is a specialized procedure that has extremely high specificity but lower sensitivity in patients with high CD+4 cells counts and low viremia. This test is only limited in reference laboratories. Antigen detection. P24 antigen detection by Western blotting is a confirmatory test of acute HIV infection. Nucleic and detection. The detection of viral nucleic acid, the proviral DNA by PCR, viral RNA by RT-PCR provides a specific and sensitive diagnosis to identify persons who are infected by hematological and serological diagnosis. Hematological diagnosis ma hemoglobin, SS, anemia before ACT, bone marrow suppression during ARV, white blood cells counts and differentials. High WBC level may signify infection. Low WBC may indicate disease, progression or bone marrow suppression. Contrimazole. C मा छ TCL. TCL is less than 1200 per ml has been correlated with CD4 count less than 200 cells per ml. TCL should not be used to make decisions on initiation of ARV in asymptomatic patients. Indirect detection. Indirect detection मा छ serological diagnosis. The Western blot detects antibodies to the following specific HIV-1 protein core P17, P24 and gap precursors P40 and P55, polymerase P31, P51, P66 and envelope GP41, GP120/160. ELISA enzyme-linked immunosorbent ELISA enzyme-linked immunosorbent assays and IFA immunofluorescence assays can be employed for serodiagnosis. For HIV-2 infection, antibodies against P26 and GP36 are detected.